Good evening, people. Good evening. Hope you can hear me clearly. Am I audible? Can you hear me clearly, people? Hello, hello. Can you hear me? Yes. Okay, fine. So, our job today, we have to discuss the paper number 12. Shall we find, can you find that paper? Paper number 12. So, yeah, let me share this paper for you. And this is the paper, ne? This is paper. Try people, yep. So let me enlarge this paper. So we'll go with the MCQ part first. Hope you have answered and uploaded. I think all the uploaded papers are marked. Right. Hope you have uploaded it. Right. Okay, people will check your answers. Question number 1, after Krebs cycle, what is the destiny of carbon atom? What is the destiny of carbon atom of pyruvate after Krebs cycle? After Krebs cycle, what is the destiny of carbon atom of pyruvate after the Krebs cycle? Ne? So what happened, people? It carbon present in pyruvate, carbon dioxide is going to be released, and then Krebs cycle, ne? Carbon dioxide is going to release. Actually, I think this question is better if they ask what is the destiny of the carbon present in acetyl part, ne? Right? What is the destiny of the carbon present in acetyl rather than asking for the pyruvate. Production of ATP from electron transport chain. Production of ATP from electron transport chain. Wrong, neither. And then release of atmosphere as part of carbon dioxide molecule. I can agree with that. And then binds with citric acid to form acetyl coenzyme A. Bind with citric acid to form acetyl coenzyme A. Citric acid to form acetyl coenzyme A, wrong, neither. We know that is not what happened. Oxaloacetate, it bind and then form citrate. So it's not correct. These carbon atoms collect at the intermembrane space, wrong. Cyclically enters to Krebs cycle. No, it's actually, there is a cyclic event, neither. But we can take, people, the best answer as the second one. Question number 2. Question number 2. Question 2. Which can be considered as an event that occur outside the mitochondria in aerobic respiration. Which can considered as an event that occur outside mitochondria in aerobic respiration. Releasing of carbon dioxide by decarboxylation. Outside in mitochondria, people, release of carbon dioxide by decarboxylation. Oxidation of FADH2. Reduction of molecular oxygen. Production of ATP by substrate level phosphorylation. Complete oxidation of glucose molecule. Production of ATP by substrate level phosphorylation, people, it can happen in glycolysis too, neither. So why don't we go for the fourth one as the answer? Right. Moving to next question. Moving to next question. Right. Moving to next question. Question number 3. Question number 3. Select the correct statement about anaerobic respiration. Correct statement regarding anaerobic respiration. ATP produced in glycolysis cannot be used by the cell. ATP produced in glycolysis cannot be used by the cell. Correct statement regarding anaerobic respiration. ATP produced in glycolysis cannot be used by the cell. Correct statement regarding anaerobic. ATP produced in glycolysis cannot be used by the cell. Wrong, neither. That's we don't agree with that. There are only 2 types of fermentation. There are 2 main types of fermentation. Neither. 2 main types of fermentation. I think I can take it as wrong. If you check the resource book, what's resource book says about that, people? It says fermentation is a method of production of ATP. There are many types of fermentation. Many types of fermentation. So only 2 types of fermentation is not a correct statement. Decarboxylation happens in ethyl alcohol fermentation. Decarboxylation happens in ethyl alcohol fermentation. What do you say about that? Yeah, ethyl alcohol fermentation. Perfect, neither. Not in lactic acid fermentation. Final hydrogen acceptor is acetaldehyde. Final hydrogen acceptor is acetaldehyde in lactic acid fermentation. Wrong. Acetaldehyde is in ethyl alcohol fermentation. Most common organism that does ethyl alcohol fermentation is bacteria. Most common organism ethyl alcohol. No, bacteria does that. Neither. It says many bacteria carry out ethyl alcohol fermentation. Most common organism which carry out ethyl alcohol fermentation is yeast. So we can take this as wrong. So we got the answer. Answer is 3, right? Moving to next question. Question number 4. Which one of the following statement is correct regarding anaerobic respiration? Following is correct regarding anaerobic respiration. In the ethyl alcohol fermentation, primary hydrogen acceptor is pyruvate. In ethyl alcohol fermentation, primary hydrogen acceptor is pyruvate. Ethyl alcohol fermentation, primary hydrogen acceptor is pyruvate. How about that? Wrong, neither. Acetaldehyde, neither. Right? End products of lactic acid fermentation are lactic acid, ATP, and NADH. End products of lactic acid fermentation are lactic acid, ATP, and NADH. Wrong, ne? NAD plus. NAD plus. End product of alcohol fermentation are carbon dioxide, ATP, and NADH. If you are weaker in this section, for your information, we are going to do it for 2027 students, right? So you can interested, you can join and then learn with the juniors. The end product of alcohol fermentation are carbon dioxide, ATP, and NADH. Why is it repeating once again? That's wrong. Every time in fermentation, the product must be NAD plus, not NADH. So that's wrong. ATP synthesized when pyruvate is converted to lactic acid. ATP is synthesized when pyruvate is converted to lactic acid. ATP is synthesized when pyruvate is converted to lactic acid. That's wrong, neither. No. ATP produced during glycolysis only. Hydrogen released from one molecule of glucose reduced 2 NAD plus into 2 NADH. Hydrogen released from one molecule of glucose reduced 2 NAD plus into 2 NADH. I can agree with that. Hydrogen released from one molecule of glucose reduced NAD plus into 2 NADH. Yeah, that's correct. Yeah, that's perfect. It happens in glycolysis, people. Glycolysis, yeah, it's given 4 hydrogen. Hydrogens kila. This says, people, 4 hydrogen ions and electrons released from the 1 molecule of glucose breakdown reduce 2 NAD plus into 2 NADH. Hydrogens hydrogens released from 1. Now, if it says 2 hydrogen, then wrong. It got to be 4. Ne? So, we got the answer here, 5. Any questions, students? Any questions? No? RQ was calculated periodically for a given sample of germinating mung seeds in a flask. RQ changes from 0.1 to 0.7 with time. RQ value changes from 1.0 to 0.7. So that means lipids involving in cellular respiration. Moving to next page. Moving to next page. Aerobic respiration is replaced by anaerobic respiration. No, it's actually from carbohydrate, it goes to lipids. Change the respiratory substrate from carbohydrate to oil. That's correct. Answer is third one. Answer is 3. All right. Question number 6. Question number 6. Which of the following response is true? Which of the following response is true? The energy released by breakdown of high energy phosphate bond is ATP is 30.5 kilojoules per mole. All high energy phosphate bonds neither. One bond neither. Wrong statement, right? And then ADP is universal energy carrier. No. ATP can be produced with living cells using ADP, inorganic phosphate, and energy. Yeah. And then substrate level phosphorylation occurs in photosynthesis. No. And then free energy released by oxidation of compounds during cell respiration is not used for ATP synthesis. Free energy released by oxidation of compounds during cell respiration is not used for ATP synthesis. Free energy released by oxidation of compounds during cell respiration is not used. Wrong neither. It's used. That's what happened in the substrate level phosphorylation. So 6 I'm taking the answer as 3. 6 answer is 3. Ne? 5. Okay, fine. So if it is different with the marking scheme, please let me know. Right. Question number 7. Some events that occur during the evolution of organisms are given below. Some events that occur during evolution. And then state the correct sequence of above events. Appearance of unicellular eukaryotes, evolution of multicellular, diversification of unicellular eukaryotes, protein similar to. I think C as the first, neither. C first. Appearance of unicellular eukaryotes, CA. Do you have CA? Ilagata multicellular forms. CABD. CABD. Seventh one, 2, 3. CA or AC. Diversification. Okay. I'm wrong. It has to be C, third one. Reason, because it says eukaryotes. So it has prokaryotes. So diversification, unicell. Yeah. Okay. ACBD. All right. Okay. You're awake, ne? You are awake, right? Good sign. Right. Question number 8. Which of the following correct regarding origin of life on Earth and evolution? Correct regarding origin of life on Earth and evolution. Synthesis of organic molecules from inorganic molecules occurred in oceans. Organic molecules from inorganic molecules occurred in oceans. You can mark these answers. That is going to be a little help for me also. You can mark neither. You can turn on annotation and then go on marking. Synthesis of organic molecules from inorganic molecules occur in the ocean. Organic molecule from inorganic occur in the ocean. No, neither. It in the atmosphere. Ne? Wrong. They are asking correct. Ne? Protocell was formed in the accumulation of RNA into lipid and protein bound vesicles. Lipid and protein bound vesicles. Yeah, correct. Tetrapod evolved from lobed finfish. Tetrapods evolved from lobed finfish. Correct regarding RNA. Protocell were formed by accumulation of RNA into lipid and protein-bound vesicles. Tetrapods evolved from lobed finfish. The origin of human lineage took place 10 million years ago. That's wrong. And then fossils of the oldest known protists are similar to brown algae, wrong, red algae, right? Tetrapods evolved from lobed finfish. Protocell was formed by accumulation of RNA. Okay. Sorry, but the balamena protocell formed the early genes enzyme would have been RNA. These protocells, RNA accumulated lipid-bound vesicles and form protocells. RNA accumulated into lipid-bound vesicles, which exhibit enzyme catalyzed activities. And proteins not proteins, lipid-bound vesicles. Then we'll go with the third one as the answer. 813. Question number 9. Yeah. Right. Question number 9. Which statement time period is not matching? Origin of photosynthetic organisms, proterozoic. Not matching, honey. Proterozoic. Haidene, Archean, proterozoic. Photosynthetic organisms, atmosphere oxygen. Archean, so it's not correct. Origin of reptiles and other radiation and their radiation, Phanerozoic, Proterozoic, this is wrong. Reptiles and their radiation, Phanerozoic, correct. Radiation of pollinating insects, Cenozoic, Hari. Origin of eukaryotic cells, Proterozoic people, eukaryotic cells, correct. And then origin of Earth, Hadean, Eon, Harine. We got the answer, first one. Right, question number 10. Which of the following statement regarding evolution and diversity of organism is correct? Evolution and diversity of organism is correct. Colonization of plant and animals on land appeared in Paleozoic era. Plant and animals on land appeared in Paleozoic colonization. Paleozoic era. Harined? Correct, ne? Asking what? Correct. Colonization of plants and animals in the land appeared in Paleozoic. Paleozoic Cam Odo. Odobishan, ne? Paleozoic era, correct. Concentration of carbon dioxide atmosphere increase in the Archean eon. We didn't say that, ne? Wrong. Origin of mammals appeared in Paleozoic. No, mammals in Mesozoic. And then Mesozoic era, origin of human ancestors, no. Origin of most present-day groups of insects appeared in Mesozoic. Present-day groups, Mesozoic, no, Paleozoic. Right, Paleozoic. Right, moving to next set of questions. Which is incorrect regarding diversification of eukaryotes? Which is incorrect regarding diversification of eukaryotes? The fossils of the first eukaryotic organisms are about 1.2 billion years ago. First eukaryote 1.2, wrong, ne? 1.8, ne? No. So first is incorrect. According to the DNA analysis, spongy evolved ekahari. Chordates evolved 670 hari. Colonization of terrestrial plant was started before evolution of arthropods. Terrestrial plants was started before evolution of arthropods. Colonization of terrestrial plants was started before evolution. Wrong, neither. Oh. The earliest tetrapods formed about 380 million years ago, which were evolved from lobed finfish. 380, 380, 380, which the emergence largely is differentiation of roots, limbs, 380 million years ago. Wrong, neither. 365 lobed finfish. 365 million years ago. Earliest tetrapods formed 365 million years ago. They are asking incorrect. All right. What is your suggestions on that? What do you say? Spongy 700 million years ago. DNA analysis spongy evolved 700 million years ago. Are spongy kidalama? According to DNA, spongy evolved 700 million years ago. Okay, ne? Chordates have evolved. That is chordate 670. Chordates had evolved 670. Origination ancestors of chordates. Are so they are asking incorrect. So we have couple of incorrect statements. Colonization to plants started before evolution of arthropods. Wrong, ne? So I got three answers here, people. 1, 4, 5. 1, 4, 5. Right, we'll go with that. 1, 4, 5. Right. 1, 4, 5. Why? So it said 7 billion. Yeah, I can. 700 billion ne? Billion wrong ne? Billion. So then what's the answer? Only third one. So then the question is wrong ne? You have to select the correct regarding the diverse equation. Correct regarding. Then we'll take people correct one. We'll correct the question. So do that correction. 11th, I have given you answer people third. Yes, third, I think then the question error. Question error. All right. So sorry about that. Moving to question number 12. The plant phylum have the largest least developed gametophytes. Least developed gametophyte. No doubt that it is Anthophyta. Least developed gametophyte is Anthophyta, my dear. Why? Because the gametophytes are well developed in Bryophyta. Least developed in Anthophyta. So we'll discuss Anthophyta life cycle next week. Right, next Wednesday night we'll discuss. Anyway, do you have suggestions for next sections? Can you suggest sections? Next sections, Anthophyta comes for sure. So what should I consider for making questions on this Saturday paper number 13? Paper number 13, what are your suggestions, students? Any suggestions? Anthophyta, okay? Anthophyta. Anthophyta. Any other? Paper 13. Anything else you would like to suggest? Paper 13. Invertebrates. Invertebrates, didn't we do invertebrates? We didn't. Invertebrates we did. Protista. I will take Protista. Protista classification part, did we take dichotomous key and all we took, ne? Right? Then Protista. Then Anthophyta, Protista. How about photosynthesis? Did we discuss photosynthesis? We did, ne? I prepared questions, ne? How about unit 2 section? Cell cycle. Didn't we do recently? No name. We did that recently, cell cycle. Cell cycle, did we do it recently? Okay, cell cycle. Cell cycle. Protista, Anthophyta. All right. Good enough. 3 sections. Right. Okay. Right. Moving back to the paper. Moving back to the questions. Question number 13. In the life cycle of Coecus, sporophyte are heterosporous and monoecious. Sporophytes are monoecious. What do you call monoecious? One house. Sporophytes are monoecious. That's correct. One house. क्या नहीं bisexual नहीं? The wrong नहीं? Oh, one house. So cell cycle also included. So cell cycle also included. Yeah, we'll take cell cycle also. Okay. Sporophytes are life cycle of Coecus. Sporophytes are heterosporous. That is correct. Monoecious. Wrong, ne? Why? Dioecious. Unisexual. Dioecious, unisexual. Are the next one, ovules are produced ventrally on the megasporophylls. No, we didn't say that. Megaspores are not released into the external environment. Megaspores are not released into the external environment. That's correct. Large number of biflagellate sperms are produced in the male plant. Large number of biflagellate sperms are produced in male plant. Large number of biflagellate sperms. Is it biflagellated? Wrong, ne? Wrong, ne? Relatively, the female plant can live longer time than the male plant. Relatively, the female plant can live longer time than the male plant. No, we didn't say that. We can take third one as the answer. Third as the answer. Question number 14. Question number 14. Which of the following responses indicate haploid, diploid, and triploid structures, respectively? Archegonium haploid, angiosperm ovule. Angiosperm ovule is a diploid structure. Coecus endosperm is a diploid structure. You can't take that, ne? Because the triploid structure is the angiosperm endosperm. So if you have any answer for the third point, that can be the answer possibly, ne? Angiosperm endosperm. Selagilla megasporangium, Neprolepis gametophyte. So we can go with the fifth one, right? So if you can be little bit logical, you can pick the answer. Common to Neprolepis, Selagilla and Coecus. So biology is actually easy, very easy, right? So we discuss papers. We have a strategy. We discuss papers early as possible, right? Not in the last moment, some people discuss papers at the last moment, in the last couple of hours. That's not a good strategy because you can't learn, right? So this is the best way of learning, right? Do questions early and then learn based on questions. So we'll continue with that. Common to Neprolepis, Selagilla and Coecus having monoecious gametophyte. Neprolepis, Selagilla and Coecus having monoecious gametophyte. Wrong, neither. Neprolepis, monoecious gametophyte, correct. Selagilla, no. Dioecious. Coecus, dioecious. No. Being heterosporous, no. Neprolepis is not heterosporous. Starch being the storage food. Starch being the storage food. Starch being the storage food. Yeah. Because they are plants, right? Presence of vessel elements in the xylem, no. Having autotrophic gametophytes. No, Coecus gametophyte is not autotrophic. So I think third is better. Third is the better answer. Yes, good. Third is the better answer. Right, moving people for the structured essay paper. Structured essay paper, hope you have uploaded it. Right, hope you have uploaded it. Right, people? Structure they say define aerobic respiration, the process of synthesizing ATP from respiratory substrates such as glucose in presence of molecular oxygen, right? In presence of molecular oxygen. Now this word molecular oxygen is heavy, okay? If you just say oxygen, then wrong. You got to say molecular oxygen, all right? So you have to emphasize on that. Reason in aerobic respiration, molecular oxygen important. Complete following equation. So how that was given to you? So pyruvate, ne? Pyruvate. Okay, so you write pyruvate here. And then what is here? Acetyl coenzyme A, acetyl CoA. So this is it. Oops. Oops. Pyruvate. Pyruvate and acetyl coenzyme A. Right. State the site of tricarboxylic acid cycle. That is the matrix of mitochondria. Tricarboxylic acid cycle, you know, you can call as the Krebs cycle, TCA cycle. So different names are there. What is the process of forming ATP in TCA cycle? ATP in TCA cycle, substrate level phosphorylation, and then state the number of reduced FAD. State the number of reduced FAD and reduced NAD formed in Krebs cycle. How many formed in Krebs cycle? Reduce FAD 2, reduce NAD in Krebs cycle. For one cycle, it's 3. For both cycle, it is 6. So this is will take people for the cycle to run 2 times. All right. So as 6. So I'll give it per glucose molecule. All right. Fill blanks to show number of ATP produced during aerobic respiration. In glycolysis, ATP2, NADH, glycolysis. Fill blanks to show number of ATP produced during aerobic respiration. In glycolysis, ATP2, from NADH, it is 5. NADH 5. Fill blanks to show the number of, let me check the question, people. Let me check the question. State the number of reduced FAD and reduced NADH formed in Krebs cycle. Reduced FAD 2, ne? NAD formed in Krebs cycle. Krebs cycle, if you take Krebs cycle alone, 3 NADH is produced. 3 NADH forms in the Krebs cycle alone. State the number of reduced FAD, H2 and reduced NAD. So what is mentioned here? 5, wrong, neither. 3. Oh my God. Glycolysis. Part B, number of reduced FAD and formed in Krebs cycle. That's 2 different questions, neither. Okay, this is part 5. Okay, let me. Okay, I think state the number of reduced FAD, H2 and reduced NAD formed by the Krebs cycle. This is okay, ne? This is okay. Okay, I answer it. In glycolysis, let me answer it once again, people. In glycolysis, fill the blanks to show number of ATP produced during aerobic respiration. In glycolysis, ATP2 from NADH in glycolysis, ne? Each NADH 2.5, so this is 5, right? Each NADH 2.5 for 2 NADH, then 5, okay? And then in pyruvate oxidation from NADH, pyruvate oxidation, 5 once again. Why? 2 NADH, so it's going to be 5. In citric acid cycle as ATP, 2. Citric acid cycle direct ATP 2. Right, moving to next page. Moving to next page. So if you have questions, ask. From NADH, from NADH, 15. How many NADH people? How many NADH? How many NADH? Fill the blanks to show number of ATP produced during aerobic respiration in citric acid cycle from NADH. Citric acid cycle, we know NADH, how many 6 is produced? Ne? 6 into 2.5, 15. Ne? 6 into 2.5, 15. 6 into 2.5, 15. Right. And then FADH2, 1.5. So from 2, 1.5, it's 2 for 3. Ne? Right. Right. Moving to next part B, set apparatus used to determine the rate of respiration given below. Write an equation for the rate of respiration according to the above experiment. Now here H1 is oxygen. Ne? So this is one way of writing the rate of respiration. H1 divided by time. Ne? H1 divided by time. One of the way of writing the rate. Or else you can write the rate based on carbon dioxide, H1 minus H2. Right? H1 minus H2 is carbon dioxide volume, right? So you divide by time. Define respiratory quotient, the ratio of carbon dioxide evolved and volume of oxygen consumed for given time for the respiratory substrate. So this is important, eh? Given time for the respiratory substrate. R cube value. Given time for the respiratory substrate. Hari. What is the respiratory quotient of seeds used? 0.7. Castor seeds 0.7. What is the liquid level B setup after 2 hours? If the germinating green gram seeds remain at 0, ne? Why? Because for carbohydrate containing food, oxygen and for carbohydrate containing seeds, oxygen and the carbon dioxide level is equal. So RQ value comes to 1. Part C1, 4 events occur during evolution of organism as follows. Right above events in chronological order. Evolution of spongy, origin of unicellular eukaryotes. RNA exhibited the enzyme catalyzed activities and were able to replicate. Water bodies became saturated with oxygen. I think C as first, ne? And then D. And then BA, right? So this is the answer. Right, question number 2. Question number 2. You have to mark people whether these statements are correct or wrong. Kingdom Plantae was first identified by Carolus Linnaeus. Kingdom Plantae and Animalia. Kingdom Plantae was first identified by Carolus Linnaeus. Yeah, okay. Ne? He introduced the hierarchical levels. So I'll take it as correct. Plants and animals level classification was done by Aristotle. But the hierarchical level introduced it as a taxon by Linnaeus. Carolus Linnaeus, this botanist. Classification, kingdom of organism, plants and animals. He identified 2 kingdoms of organisms, correct? Ilangata, Protista is the natural kingdom where organisms with different evolutionary origins are included. Protista is the natural kingdom where organisms of different evolutionary origins are included, correct? correct anyway, wrong, why? Natural kingdom wrong, artificial. The number of common characteristics found within the genus is higher than that of the species. Wrong, species has higher. Eukaryotic and archaea are most related to each other than bacteria. Eukarya and archaea are most related to each other than bacteria. Eukarya and archaea are most related to each other than bacteria. And then people state the major events that took place in evolution origin during the following time period. 1.8 billion years ago, first eukaryotic appeared, Hari. And then about 2.7 billion years ago, photosynthetic organisms, cyanobacteria. Name the kingdom included domain archaea. Kingdom included domain archaea. Archaebacteria, okay, but that's not mentioned in the resource book anyway. We'll take it as archaebacteria. Write the scientific name of the following fruits with two wings: Dipterocarpus zeylanicus, human, Homo sapiens. So when you write, you have to write the first letter of Dipterocarpus in capital, zeylanicus properly spelled, separately underlined. Those things must be there. Your answer you can't text. Okay, check for the nomenclature rules. You have to follow them. All right. Then people moving to next question. Following diagram shows life cycle of a plant. This is life cycle of Selaginella. Identify the plant Selaginella. What is the phylum Lycophyta? State another name for the group of plants. Seedless vascular plants. I think you can write also as people spike mosses. Spike mosses or maybe seedless vascular plants, fine. Label A to I. A to I, A sporophyte, B strobilus, C microsporangium, D megasporangium, E megaspore, I flagellated sperms, G egg, H diploid zygote, and finally I young sporophyte. Hope you have done that. It's not a hard part. We have discussed this life cycle recently. What are heterophilous plants? Plants with two types of leaves, megaphylls and microphylls. How many megaspores are formed in megasporangium? 4. State features of female gametophyte of above plant. Multicellular, surrounded by thick wall of the megaspore. Few rhizoids are present. Yeah, gametophyte of the above plant. Female gametophyte is microscopic anyway. Microscopic structure, right, covered by the megaspore wall. Part B1, what are features of seed plants? Production of seeds, reduction of gametophyte, heterospory, production of ovules and eggs, production of pollen grains. Ficus plant is more adapted than Selaginella for land life. Compound leaves show xerophytic adaptation. Young leaves are fiddlehead. What are different features of male and female sporophyte of Ficus? Male sporophyte produce male cones with the microsporophylls. Female sporophyte produce crown of megasporophylls. Label following diagram. Right. This diagram is showing you Ficus ovule. So A is micropyle, fine, B is integument, correct. C is pollen chamber. Oh my God, this is wrong. Right, people, we have to make a correction. This one, A is not a micropyle. That's the pollen chamber. This one, pollen chamber, lamai, pollen, I can't write. Pollen, chamber. I'll say pollen chahari. Pollen chamber. B is integument, okay. C, pollen chamber. What is that, people? Yak. C is archegonial chamber. C is archegonial chamber. Please correct. Archegonial chamber. Archegonial. C is archegonia. What is D? D is branched pollen tube. That's correct. E is archegonium. Archegonium. And F, endosperm, correct. So do that correction. I hope you have done it properly because you are better than me, right? My students are always better than me. State how fertilization occurs in Caecus. Fertilization occurs in Caecus. The end of the pollen tube ruptures and release sperms into the archegonal chamber of the ovule, and sperms swim into the liquid medium and then fertilize the zygote. That is okay. Part C, people, write in words and arrows a summary for life cycle of Selaginella and Caecus. Life cycle of Selaginella. This is my summary. Hope you can write that now. Sporophyte people, can you write there 2N kiladaganlamai brackets volta? 2N strobilus, microsporophyll, megasporophyll, microsporangium, microspore mother cell. 2N, the microspore mother cell, mark the ploidy level, 2N. And then microspore. Here you had write, what are you going to write here, my dear? Meiosis, ne? Look at my handwriting. I'm writing with the mouse. You can't write. Microspore, male gametophyte, sperm fine. And here megasporangium, megaspore mother cell, 2N, dear. Put it as 2N. Harida, boys. Female gametophyte, megaspore. So here, meiosis once again. Meiosis. So that's the life cycle. Hope you have completed that life cycle. Then people, life cycle of Caecus. Caecus life cycle. Caecus. Right, this is life cycle of Caecus. So we start with the male sporophyte, microsporophyll, microsporangium, microspore mother cells, meiosis here, microspore. Here put the ploidy number. Male sporophyte 2N, microspore mother cells 2N. Microspore is haploid, all right? Haploid, kiladanna, male gametophyte, sperm, hari. They say female sporophyte, megasporophyll, megasporangium. Where is ovule? Megasporophyll, megasporangium. I think you can write here. Ovule කියලා ලියන්න පුළුවන්. Ovule here mention නේද? Megaspore mother cells. Megaspore. Here meiosis. Here meiosis. Okay, I think you can do that. Archegonium, egg, zygote, embryo, seed. Yeah, the cycle starts over once again. Then people, the key difference between sporophytes of these two plants, Selaginella, herbaceous plant, not a herb. Caecus, not a herb, it's a big tree, right? Heterophyllous leaves are absent. What is heterophyllous? Small and large leaves are there. Stem is dorsoventrally flattened, stem is columnar, tall tree. dichotomously branched roots, taproot system present. Monoecious, dioecious. Which one? Sporophyte. So you write monoecious sporophyte. Secondary growth absent, secondary growth present. Okay, done. Then people, how female gametophyte of Selegina differ from that of the Caecus? Female gametophyte of Selegina differ from Caecus. Female gametophyte of Selegina partial depending on the sporophyte, but female gametophyte of Caecus depends on the sporophyte. So no partial, fully dependent. Female gametophyte of Selegina develop within the strobulus. But female gametophyte of Caecus develop within the ovule. Female gametophyte of Selegina develop within the strobulus. Female gametophyte of Caecus develop within the ovule. Okay, fine. Because female gametophyte takes some nutrients from the sporophyte. Female gametophyte of Selegina are released to the external environment, but female gametophyte of Caecus is not released. All right, people. Let us state with the following statements. State with the following statements are correct regarding Selaginella and Caecus. Remaining female gametophyte develops into endosperm in both plants. Remaining female gametophyte develop into endosperm in both plants. Wrong, neither. Why? Selaginella, you can't take it as correct. Wind dispersed spores are present in both. Wind dispersed spores are present in both. Oh, yeah, correct. It must say female gametophyte is photosynthetic in Selaginella and not in Caecus. Female gametophyte is photosynthetic in Selaginella, not in Caecus. Harine. Mature gametophyte grow in external environment. Mature gametophyte grow in external environment. What do you say about that? Incorrect. Why? Wrong for Caecus. Right, people, moving to next page. Moving to next page. Right, then we have to start SAB part. SAB. I think it's a good paper, good paper, because it covers some important theory parts. Questions are also good, quite moderate questions, not too easy, not too difficult. Right, question is a brief describe properties of sporophyte and gametophyte of Selaginella and Caecus. People, Caecus, Selaginella, gametophyte and sporophyte. So I have just included points in the resource book. The sporophyte details first and gametophyte details second, right? Selaginella sporophyte. Now, when you say sporophyte, you can't like put a border, but so sporophyte, I have given marks like describe in the root, stem, leaves, and then the strobulus and the microsporophylls, megasporophylls up to formation of the spore, right? The sporangium details I have given marks here, right? Up to that, I have included marks for that, the sporophytic details. Then up to this point, right? So, right, is the sporophyte details. Then after that, the gametophyte details. Gametophyte details of Selaginella. Okay. Then people, yeah, up to this, the gametophyte. Selaginella, there are two gametophytes. Male gametophyte is there, female gametophyte present. So I have included marks for them both. Some people said that they didn't understand the question. That is like quite simple. When you understand the life cycle, if you understand the life cycle, you can put the borders, because the question is not to describe the whole Selaginella life cycle and the Caecus life cycle, right? You must write only the relevant information. Then people seek us sporophyte details. Seek us sporophyte details up to this point. Sporophyte of seekers. Remaining part is the seekers gametophyte details. You know, male gametophyte and the female gametophyte. Two gametophytes are there. So I included those points for that. Female gametophyte, pollen grain and the germinated pollen is the male gametophyte, right? So that's the answer for the question. Hope you answered it. Anyone had any question, people, regarding that the essay question? It's anywhere a model question, not a past paper. So what will happen if we just fully write both life cycles? No, that's not the question either. I think it's not suitable. It's not suitable because the question clearly says to describe the properties of sporophyte and the gametophyte of Selaginella. So why do you want to write the entire life cycle? Right? Then you don't really consider the question. You just simply go on writing everything that you know. You have to answer the question, and that's not a decent answer then, right? Describe anaerobic respiration. Take the same here. I'm asking you to describe the anaerobic respiration. You write everything that you know. That's not good, right? Aerobic respiration, Krebs cycle, electron transport chain, everything you write. On one hand, it takes unnecessarily time. You know, that question carries 38 marks points. So when you write both life cycle, definitely you can't write that within the planned time period, given time period, you won't be able to answer it. So that's weak, ne? If you can't finish it within the given time, people, then it's not a suitable answer. You have to go home and write if that happen in the exam, ne? So that's not good. So you have to understand the question and then write only the relevant things. I know this A-level is challenging when you write essay answers. You have a challenge. That I understand, right? You don't know what to write, right? So you have to use your brain. Now, when you're writing essay questions, you have to write the relevant points. It doesn't matter that you write in your own words. You can grab marks by doing that. If you remember whole resource book phrases and all, better, right? But still you can get marks, right? If you answer the question, right? It's not something difficult when somebody asks you to describe the sporophyte of Selaginella. It's not difficult for you to pick some points, ne? Stem, root, leaf differentiation present. It's a seedless vascular plant, and then you can see that the heterophyllous leaves are present on the stem, roots present, dichotomously branched, and then the strobilus present, the microsporophylls clustered, megasporophylls are clustered in the strobilus, cone-shaped structures. I think you can build up the answer, right? You can build up the answer, all right? Right, first step of this glycolysis, the first step of this is also glycolysis, anaerobic respiration. So my answer is describe anaerobic respiration. You are describing glycolysis, and then you are describing the two fermentation processes, the ethyl alcohol fermentation and the lactic acid fermentation, right? So this is the answer for that. Right. So please read the given scheme. This is carbon dioxide, my dear. Carbon dioxide, okay. And then second part of the question is to explain how early heterotrophic anaerobes have evolved to aerobic organisms. We learned that early organisms were heterotrophic anaerobic organisms. And then the around 2.7 billion years ago, photosynthetic prokaryotes had evolved. They start releasing oxygen to the seawater, which oxidize the dissolved iron, and then gradually increase the oxygen level in the atmosphere. This increase the atmospheric oxygen condition, right? And then had accelerated the origin of the chloroplast and other aerobic organism. I think in the resource book, they only talk about this. I included this part, and later on the aerobic organisms had evolved on Earth, right? So that's the answer. for the paper 12, right? So thank you very much for your participation, right? You can see very few people are supporting this sort of paper discussion. That's normally, that's how it goes, right? Everyone don't, they just come and go for classes. They're not really having that desire of going for the aim. So you have it, so you are so lucky, ne? It's I mean, no one can give it. It comes to you, ne? Right? When you try to stick, when you have your what do you call, plans, ne? Future plan and all, then you work for that, and then you like what you learn, ne? And then you want to improve yourself, and then you participate in this sort of work, ne? So there is a saying, ne? What the saying says? You can take the horse to water, but you can't make the horse to drink water, ne? Right? We can take you to water, and then some people are drinking, ne? You all, these 19 people. So I'm so happy, right? You are in the right track. So we'll continue this. And thank you very much for your time. And I'll see you all soon in the Saturday class. Okay, then. Bye-bye. See you.